next speakers are Simon Watkins and Alan Watson. Simon or Alan, are you all available to share your screen?
Yep.
All right. Perfect.
Okay.
Simon and Alan come to us from the University of Pittsburgh. Simon Watkins is a distinguished professor and chair of the Department of Cell Biology, or vice chair of the Department of Cell Biology. And Alan is a research assistant professor in that lab. And they're both leading researchers in developing cutting-edge optical imaging technologies. And the title of their talk today is Technologies for High-Speed, High-Resolution Imaging of Large Biological Systems. I'll let you take it from there.
Sure. Can you hear me, guys? I had to change computers in the middle.
Yes, you sound great.
So we've been trying to develop, this is a tool set talk really, we've been trying to develop tools which allow us to probe cellular molecular events at very high resolution but in very large tissues and essentially whole organs within animals and humans. The approach we're taking is using a confocal method because the light sheet approaches have become very popular, don't allow us to really look at very big samples. Essentially, as you all know, confocals use a pinhole to reject out of focus light, and so you get this pretty image instead of this blurry thing. And where light sheet can only be used to image very, very small things, essentially light sheet uses an orthogonal axis illumination and you detect this way. Confocal has no lateral limits at all to what you can image using it. The other thing that happens with a regular microscope, let me just, sorry, I got to turn off that laser pointer, is that a normal microscope will, if you want to take a series of images, it uses a point and shoot approach, whereas the approach we've developed uses, or the approach we're working on, uses a continuous scanning approach where it does not stop scanning. It just goes on and on. And essentially scans entire ribbons. This, we published this last year. The speed is about 30 times faster than a normal microscope. So, and it's getting faster depending on the way we use it. So what would take a day with this device would take a month with a normal confocal, or equally something that would take a week with this device would take better part of a year with a normal microscope. So this was published. The other thing that you need to do if you want to image whole organs is to image at great depth. Sorry, I'm having a little problem with the movies here. Image at great depth if we clear the tissue. So clearing has been around for a while. It's been through many, many iterations, and we've developed our own tools that we use all the time for clearing, and they're very much directed towards the kinds of questions that are being asked in this particular group. Essentially, the methodology, we call it KidClear, and it's a hybrid of the two other main tools that have been, approaches that have been developed by folks. This was developed by Alan, specifically for our kidney imaging projects. And we're now finding it works with nearly all our tissues that we throw at it. So we're using it for everything now. It gives great retention of color from fluorescent proteins, but at the same time, we get really good antibody penetration and labeling for doing very large data sets. So how do we image really deep? So for those of you who are interested, the resolution of a light microscope is actually defined by this angle here, which is the numeric aperture of the system. And as you go deeper and deeper, essentially that angle decreases, and with it, the ability to get high resolution decreases. So we were working with Nikon on the solution because there is a very simple solution. You just make really, really big objectives. And this is, so instead of having this size, you've now gone to this size, literally. And this is my hand, I won't make any jokes, holding the new objectives, which this is not the prototype, which was the final version, which was released last year. It actually has a working distance of a full centimeter. This is 8.2 for IP reasons, I think would be the best way of putting it. And it can be used with various transition fluids, which we won't get into now. So we can go deep. We can image at very high resolution, essentially at 350 nanometer resolution, around 300 to 350 nanometer resolution. So what can we do with it? This is an example of a simple project where we had a 2 centimeter by 1 centimeter by 2 centimeter piece of human kidney, and we can image at subcellular resolution and reconstruct the entire tissue. This work was done with Sonny Sims-Lucas and Jackie Ho, who are in our children's hospital here. We've actually imaged entire chunks of kidney. So this is a piece of cleared human kidney. And you can see this is a small area from it, and here's a glomerulus from that small area of it. Oops, sorry, let's go back. This is a couple of images of the devices working. I don't know how many of you have used a confocal microscope historically. I'm sure most of you, or a lot of you that have bench research have used one, but they're painfully slow to work with. These guys, as I said earlier, are much, much faster. So let's look at a few examples of what we can do with this. This is a whole mouse kidney, a neonatal mouse kidney, and we imaged the blood vessels. You can see the glomeruli. We could zoom up on this and very clearly see the substructure of the glomeruli. You can see all the structure within the glomerulus over time in this 3D object. It's this is a relatively simple data set. The other thing that's important to remember is because we have the high resolution and high signal to noise, we're able to actually find very rare events within tissues. So this experiment I'm about to show you is a total failure from the scientific point of view. We were looking at putting viruses into cells and then tracking the green. The red's absolutely unimportant here. But if you look at this, in actual fact, we're able to pull out, there's only one neuron that was actually transduced in this sample. And here we're able to pull out and map the entire arboreal architecture of that single neuron pretty handily with this approach. I think if I go out just a little bit, so there's the neuronal cell body. But if you look here, we can now actually, using some 3D image processing tools that we have in-house, we can map the entire architecture of that single neuron in the entire brain. So the resolution is there, and also luckily the selectivity is there. This is what we've done with Alison Bath at Carnegie Mellon, and is part of another funded effort where we're trying to build correlative approaches. But you can see how selective it is as that neuron vanishes into the distance. Here's another example where we can measure, the point of this is, while that last movie shows that we can pick out single events in very large tissues, this shows the ability to image everything in very densely packed tissues. So we're looking at a SOX10 is green, and the red signal is humacy. And this is what we've done with Michelle Sutherland-Smith at Vanderbilt. And I'm going to skip forward in the sake of time, but if you look here, we can actually see every single one of these little cells, and then we can actually extract, using image processing, every single cell body and count them in 3D space. I'll talk a little later about the data processing we built in, but this is actually now imaging every single one of the positive cells in the entire mouse embryo in 3D space. We can do this in any 1 centimeter volume and depth, and XY it can be up to probably 10 centimeters in each axis. We're only limited by the motion range of the stage. So here you can see every single cell body in that entire 3D volume. More recently, we've been doing work with another team in Melbourne, Australia. In this case, looking at the red color here is smooth muscle actin. The green is some sensory neurons, and this is a rat bladder. And you can see, well, I'll go forwards a little bit here again, it's for the sake of time, but you can see very clearly we can easily map both, the neurons are not very brightly stained, so there's a bit of bleed through from the green, from the red channel, which is the smooth muscle actin. But we can track these in 3D if we need to, and follow them. This is what we're happy with what we're able to get out, but we're still improving this particular technique. Perhaps this is a sort of a very nice data set because it shows multicolors in a chunk of spinal cord. This is L2 through L4 spinal cord in a rat, and we're able to rebuild the entire neuronal system and map it out in 3D space in an entire chunk of spinal cord. We have done whole spines and bits from mice and rats previously, but this is one of our more recent data sets where we're able to map all the neurons or all the motor neurons. And we're also able to, you can see here the architecture within the animals, and we're able to get penetration right through the entire tissue, which made us very happy. Obviously, you can see the resolution is just fine. We can also see all the cell bodies. The tyrosine hydroxylase shows us all those. And we can see this in 3D in 2 colors. So this is actually an orthogonal, 2 orthogonal planes, but gives you an idea that we can image very deep, but also very high resolution things, and also reconstruct everything in 3D space. We have been in or semi-closing, we have been combining this technique with expansion microscopy. Essentially, expansion microscopy moves things further apart such that you can use regular confocal microscopes to quantify and measure things at a super resolution level. This is done with a new faculty who Alan is collaborating with at Carnegie Mellon, Lian Zhao. And essentially you stretch the tissue and then you put it in a microscope. This is a really lovely data set from which maps the entire nervous system of a zebrafish embryo at 150 nanometer resolution in 3D. And with this stuff we're preparing for publication as we speak. So the other third or the fourth part, beyond resolution and the ability to image deep and image large samples, is data handling and data management. Right now, a 4-color brain at very high resolution for a whole brain is about 4 terabytes, which isn't that bad. At those resolutions, our data, it's about a petabyte a year of data. But if once we start pushing to where we're really going to, which is really using the systems at their spatial limit, we can be expecting to generate up to 250 terabytes per sample. So we're using data quite quickly. We have close collaborations, as you know, with the guys at Carnegie Mellon, Alex Mopolowski, who we have a collaborative grant to do this part of the data thing with. But right now we're up to about 2.1 petabytes, and we have some serious ability to share the data and move the data around. We're about to build another server closet, which will hold another 2 to 4 petabytes. These red boxes hold 650 terabytes each, and we're going to stack an entire one of these units with these guys here. There's also a lot of quite serious computing that we've built, but we're very dependent on the supercomputing center, the Pittsburgh Supercomputing Center, for some of that. But those interactions between us and the PSC have taught us a great deal about data traveling and moving data around. We locally have benefited a great deal from the Bitcoin fantasy and have built these devices that have many, many graphics cards, so we can do a lot of stuff very quickly. But we have very specific and very special ways where we can move data between groups. We have, as I said, we have collaborations now which involve these technologies with folks in Australia, in Germany, and several locations throughout the US. And so we've had to develop decent tools for doing this. We have an internal networking infrastructure which is 40 gigs per second. And we use things like OneDrive and Box for sharing data, but we also have several machines in our new server area where people log on remotely in Australia or Germany and then work with our very high-end machines remotely to look at the data we've generated for them. So overall, we've developed a platform which is able to image at very, very high resolution, is able to image very deep, and is able to image very large structures. And we feel that the questions that have come up throughout today, actually, many of the questions, if we put them in the framework of true, very high resolution structure, could benefit greatly from the approaches we've built. Just to thank the guys here, Jackie and Michelle at Vanderbilt, Janet in Melbourne, and there's a couple of others in Europe. And then my team here with Alan is the leader of this. Alan is really the lead on this. And then there's the rest of the other faculty within the center. And with that, I shall stop and take questions or whatever you want to do next. Thank you.
Thank you for a great talk. That was really amazing.